Welcome to Success Blueprint for JEE and NEET. Join the class 12th Biology.
Study at your own pace and save your time and energy. Please like and subscribe for more educational videos.
Introduction.
Look at each other and list 3 physical characteristics that make you different from each other.
My eyes are black and hers are brown. I am taller and our nose shape and hair color are also different.
Does your eye color or nose shape match with your parents?
Yes, my father's eyes are also brown.
What essentially makes us different?
The DNA.
Correct. Today we will learn how does DNA creates its copies to get transformed and expresses same characters in the parents and their offsprings. Objectives. At the end of this lesson, you'll be able to understand the process of replication and its importance, understand the semi-conservative nature of RNA replication, identify genetic code, understand the process of transcription, and finally, understand the process of translation. What is DNA replication? DNA replication refers to the process of copying a DNA molecule. DNA replication should occur before cell division during the S phase of the cell cycle. In double-stranded DNA, replication is possible because each strand serves as a template for a complementary strand. Michelson and Stoll experiment on semi-conservative DNA replication. Semi-conservative replication was experimentally confirmed by Matthew Michelson and Franklin Stoll in 1958. They grew E. coli in a medium containing 15NH4Cl to label the basis of DNA and then transferred some of the cells to light nitrogen, 14N, so that the newly synthesized strands would be light. They isolated DNA from bacterial cells and centrifuged it to separate DNA. After one division in light nitrogen, DNA molecules are hybrid with intermediate density. After 2 divisions, DNA molecules separated into 2 bands, one for light DNA and one for hybrid DNA. This confirmed that DNA replication is semi-conservative. Mechanism of replication. The process of DNA replication begins at the origin of replication during S phase of cell cycle. DNA strands unwind and unzips at the replication fork by enzyme helicases. As a result, 2 separate strands are made available to act as templates. DNA polymerases uses a template to catalyze the polymerization of deoxynucleotide in 5' to 3' direction only. Thus, continuous replication occurs on one strand with polarity 3' to 5', and discontinuous replication on the other strand with polarity 5' to 3'. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. Transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Gene in the transcription unit. A gene is defined as a functional unit of inheritance. Cistron is defined as a DNA segment that codes for polypeptides. In eukaryotes, structural gene is monocistronic. In prokaryotes, structural gene is polycistronic. Monocistronic genes have interrupted coding sequences in the form of exons and introns. Exons. Exons are said to be those sequences that appear in mature or processed RNA. Introns. Introns are the intervening sequences do not appear in mature or processed RNA. Types of RNA. There are 3 types of RNA required to synthesize a protein in a cell: mRNA, tRNA, and rRNA. mRNA, or messenger RNA, provides template by carrying coded base sequence from nucleus to ribosomes. tRNA, or transfer RNA, carries amino acids to the ribosomes, reads the genetic code. rRNA, or ribosomal RNA, plays structural and catalytic role during translation. Process of transcription in bacteria. The process of transcription involves the following steps. Initiation. Initiation factor helps in the initiation of transcription. RNA polymerase binds to DNA at the promoter. DNA helix is unwound, exposing the template strand. Elongation. mRNA is synthesized using one strand of DNA as a template in 5' to 3' direction. As elongation proceeds, RNA polymerase moves along the DNA, synthesizing mRNA. The previously transcribed DNA rewinds. RNA reaches the termination sequence at the end of the chain. Termination. Using termination factor, transcription terminates. mRNA and RNA polymerase are released. Process of transcription in eukaryotes. Along with the basic transcription process, 2 additional complexities are associated with transcription in eukaryotes. Presence of 3 RNA polymerases in the nucleus. RNA polymerase 1 transcribes rRNAs. RNA polymerase 2 transcribes hnRNA. RNA polymerase 3 transcribes tRNA, 5srRNA, and snRNAs. Presence of exons and introns. Introns are removed from the genes by splicing followed by capping and tailing of hnRNA. Assessment. Before proceeding further, let us know how much you have learned. Genetic code. The genetic code is the set of rules by which information encoded in genetic material, DNA or mRNA, is translated into amino acid sequences. The code defines how sequences of 3 nucleotides, called codons, specify which amino acid will be added next during protein synthesis. Salient features of genetic code. Codon is a triplet. 61 codons code for 20 amino acids and 3 stop codons do not code for any amino acid. Each codon is unambiguous and specific for one amino acid. The code is nearly universal among all living organisms. The codon for methionine, AUG, also acts as an initiator codon. Effect of mutation on genetic code. Effect of point mutations that inserts or deletes a base in structural gene can change the amino acid sequence. Example, in sickle cell anemia, a single base pair change in the beta hemoglobin chain results in change of amino acid sequence from glutamate to valine. tRNA. tRNA acts as an adapter molecule that reads the genetic code and simultaneously binds to specific amino acids. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acids. The secondary structure of tRNA looks like a clover leaf, while in actual structure, the tRNA is compact molecule which looks like inverted L. Translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. The cellular factory responsible for synthesizing proteins is the ribosome. Ribosome consists of two subunits in its inactive state, small and large subunits. There are two sites in the large subunit for subsequent amino acids to bind to and bring them close enough to each other for the formation of peptide bond. A translation unit in mRNA consists of start codon AUG, stop codon, untranslated UTR regions. Steps of translation. The first phase of translation is known as charging of tRNA or amino acylation of tRNA, which provides energy for peptide bond formation between amino acids. When the small subunit of ribosome encounters an mRNA, it stimulates large subunit to bind to the mRNA at the start codon AUG that is recognized only by the initiator tRNA. Each time a new tRNA comes into the ribosome, the amino acid that it was carrying gets added to the elongating polypeptide chain. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Did you know? Molecular combing is a technique that allows the quantitative analysis of DNA replication. In this method, chromatin from a specific nucleus is differentially labeled at successive time points after the beginning of DNA synthesis. Genomic DNA is then extracted and combed on a glass surface, and direct measurements are made on the labeled DNA. Summary. Let us summarize what we have learned. The DNA replicates semi-conservatively. The process is guided by the complementary H bonding. During transcription also, one of the strands of DNA acts as a template to direct the synthesis of complementary RNA. In eukaryotes, transcription is accompanied by splicing of introns. The messenger RNA contains the base sequences that are read in a combination of 3 to make triplet genetic code to code for an amino acid. The site of translation protein synthesis is ribosome, which bind to mRNA and provide platform for joining of amino acids.
Friends, welcome to Kwiiz Success. Are you tired of memorizing without really understanding concepts for exams? Our innovative learning techniques help students grasp concepts easily and effectively. Our Success Blueprint course for class 11 and 12 gives you a real exam-like experience, helping you build confidence and score higher in IITG and NEET. Here, you'll find the perfect combination of 3D animated videos, AI-powered test series, and ebooks enriched with MCQs and mind maps. All our courses are designed by IIT and IIM alumni to ensure top-quality learning. For online courses and ebooks, visit www.success.org.in. Download the Success Kwiiz mobile app for free. For advanced test series and free mock exams, visit www.kwiiz.success.org.in. Download the Kwiiz Success mobile app and get a real improvement in your scores. Success is now in your hands. Thousands of students have already benefited from Kwiiz Success. Now it's your turn. Study from home, learn at your own pace, and prepare at your convenience. Don't forget to subscribe to our YouTube channel, Kwiiz Success, and share it with your friends. Thank you, and all the best for your success.